Great. Okay, well, so I will continue my tradition of being a troublemaker. And what we're going to do is we're going to start actually, I'm going to point out something I think that's really important. Epistemology, what in the world? Well, it's actually a philosophical field. It's a branch of philosophy. And I'm going to point out that all of us who get our PhDs are actually getting a doctor of philosophy, even though we tend to want to forget that. So today, you're going to have to put up with about 10 or 15 minutes that proves that I have a PhD versus something else. Epistemology, what in the world? Okay, it's one of the major branches of philosophy, and it comes from the ancient Greek root of episte, which is the knowledge, and it's how philosophers think about and try to answer the question, how do we know things? And at what point does a belief become a truth? And when are beliefs justified? And so it also includes some other subtopics like, what are the limits to knowledge? At what point can't we know things? And at what point must we just rely on belief? And, you know, what's the role of psychology on perception and on memory and testimony, which is basically, testimony is not just in a courtroom where people are saying, I saw somebody do something wrong. That's what we're doing when we collect data. We're also making eyewitness testimony. So the issue is that this is actually kind of important stuff because it's the root of the data that we analyze, because we want to believe that what we're doing is truth and not just our own personal feelings. And the issue then becomes that we as scientists owe our existence in a modern sense to a revolution in epistemology that happened 600 years ago. And it was, you know, at the dawn of the Renaissance. It's sort of as we left the Middle Ages behind, the main academic pursuit of that time was called Scholasticism. And a lot of those traditions have maintained themselves in our venerable institutions. And the thing was that scholasticists, the scholastic tradition was maintained inside of church-related structures, often monasteries. And they became their own universities at some point. And you'll notice that almost all medievally founded universities were associated with some particular religious order. And it was an attempt by people of that time to try to be academics and try to, you know, not just blindly follow everything. But in this case, what they wanted to do is they wanted to reconcile their reason to their faith that was overarching at that time. And basically what they were trying to do is fit Aristotle's views of the world and statements and dogma to Christian dogma. So they had two different dogmatic approaches, and they were trying to fit them together. And how did they fit them together? Well, they did it by making witty arguments to each other. They did it by disputing everything at great length and in minute detail. So I guess we haven't really changed that much in the last 600 years. At least those who have sit in on you know, departmental meetings can say that. But the most important thing was that scholasticists used as their idea of epistemology that knowledge was to be found in prior texts. They would find written words, and they would take those words as real, and they'd figure out how to make different texts with different statements of reality fit together. And basically what they were going to do is they were going to appeal to the authority. If something had been written down, it must be true. Well, whoops, what am I doing wrong here? There we go. Nope, wrong way. Here we go. So, where does science begin? Science begins when people begin to reject the scholastic approach to epistemology. People like Leonardo da Vinci, who say that if you find that your own experiences contradict what some authority has written, you have to trust your own observations. You have Sir Francis Bacon, saying that if instead of looking into our own minds, we had consulted experience and observation, we would have the facts and not opinions to reason about. And so we had people then, the start of modern science are people saying, no, we aren't just going to read old dusty texts and take them as truth. We're going to make sure we're going to force those observations to be tested by our own brains and by looking at the world ourselves through our own eyes. And that's the revolution that took place. And it's an epistemological revolution because we're changing what knowledge is. And with that in mind, we can take a look at, the very first modern scientific society that was founded was the Royal Society of London in 1660. And within 3 years of their founding, one of the fellows in the academy, John Evelyn, chose the Latin phrase Nullius and Verba as their motto. And it means on the word of no one, or take nobody's word for it. And according to the Royal Society, it represents the determination of fellows to withstand the domination of authority and to verify all statements by an appeal to facts. So what we have now is not people debating how many angels can dance on the head of a pin based on something that Thomas Aquinas wrote 500 years before. What we have are people saying, let's gather data. Let's see what the data tells us. So it's a profound revolution in how we know and how we assess truth. But there's a major problem here. It's something that they had to deal with early on. And that is not all observations are created equal. In fact, every one of us experiences the world slightly differently because of our psychology, because of our history, because of the way we were brought up. And so because of that, there are a number of psychological biases that can influence the observation of the world and can make empirical observations not the same. So what are some of the things that cause this? Well, different personal histories, different things that we were exposed to in our life might cause us to interpret the world in different ways. There's something called Gestalt reconfiguration, where based on our personal history, we might see random things out there in the world and connect them together incorrectly into single objects, when in fact they were just single random things that were out there. There's also sociocognitive factors. Society can influence how we process data and how we observe data. So the society around us can affect us. And this can lead to what's called cognitive dissonance, if we believe a faulty idea and we believe that it's true, when we are confronted with that, we will defend often the faulty idea with increasingly incoherent arguments, especially when we're vested in those false ideas. When we're getting something out of the false ideas, we won't let them go. So the important thing to remember, as Matthew Sharps mentions in the Skeptical Inquirer 2018, is that scientists, that we are subject to the laws of perception and cognition, and it is important for all scientists in all disciplines to be aware of these facts and to use caution in the interpretation of what might otherwise be interpreted as a purely objective observation. Now, what's he referring to? He's referring to this guy, Percival Lowell, a well-known astronomer from the late 1800s, early 1900s in the USA. He was born in Boston from a wealthy industrialist family. He went to Harvard, got a PhD in mathematics, and then went back and ran his family's businesses for 20 years before he retired early, took his money, and started his own observatory. He followed his dream to be an astronomer, went to Flagstaff, Arizona, where it was thin air, high up in the mountains with little moisture, and he built an observatory. And one of the things he got fascinated in was the planet Mars, because when he looked at Mars through his big telescope, he saw non-natural features. He saw canals, he saw oases, and he came to the conclusion that what he was looking at was massive planetary-wide engineering done by an alien race who was trying to take water from the poles and distribute it across a desert planet to allow themselves to live. Great idea. Science fiction writers grabbed onto it and have been writing stories about Mars ever since. Based on this, Ray Bradbury, Heinlein, I mean, you name the mid-20th century science fiction writer who wrote about Mars, it's always about canals. But the problem is that when other astronomers on other telescopes looked at Mars, they couldn't see those canals. Not all of them. And the problem is that you had then two groups of people arguing with each other. Lowell said, I see canals. And someone else says, no, you don't. Yes, I do. No, I don't. Yes, I do. No, I don't. And there was no way to rectify this because we did not have the technology to take photographs through telescopes at that point. And so it was just an argument about, yes, I do, no, you don't, yes, I do, no, I don't, until finally we began to get the photographic ability to take pictures. And when those pictures came in, his idea of Mars was not being replicated. And finally, in the 1960s, when the first space probes got sent to Mars, well, there were no canals of any size. Big or small, no oases, no water. It's a dusty, dry planet. And so Lowell was wrong. But how could he observe things that didn't exist? What's going on here? He was a trained scientist. He was a Harvard graduate in mathematics. He was a logical person. And we take a look at those four features. His personal history. His family's wealth came from his industrial family. Their wealth came from sending goods to and from other cities in the northeastern US by canals. They made their money from canals. The Suez Canal and Panama Canal projects had been initiated in his lifetime, and these were seen as some of the most amazing examples of human ingenuity that had ever been, created. And so he had canals in his head. Gestalt reconfiguration. He saw individual areas. And let's see if I can use this. Here we go. So this is Syrtis Major. There it is. So there's his drawing of Mars, Syrtis Major. Here's the closest image I could get from the Mars Global Explorer. And the thing is, you can see there's a fairly decent overlap in some features. But what you can see is that he was connecting random points and drawing lines between them. And that's Gestalt configuration. He saw things, he was putting together independent points into single figures that didn't exist. The sociocognitive angle, well, he got really famous from this. A lot of people knew of Percival Lowell because he was talking about aliens and he was talking about canals, and he got famous from it. And this led to cognitive dissonance because when he finally began to be confronted with people saying, what are you saying? That's not there. He took it as a personal insult. And he lashed out against people. So his trusted assistant, number 2, at his observatory in Flagstaff, he fired on the day that the guys questioned him about his canals. The guy, A.E. Douglas, says, "Are you sure you're seeing canals?" He got fired immediately. Okay, so this is how these things happen. So here's the problem. Just being Galileo or a Sir Francis Bacon and saying we have to observe the world with our own eyes, well, we have to also be a bit suspicious that our eyes are seeing things correctly. So how do we deal with this? And one solution that our forefathers in science came up with is that we need to replicate our observations. And we replicate those observations across multiple individuals and also multiple data streams. So if we can show that a pattern exists across multiple observers or multiple data streams, multiple signals, then we're going to exclude some of the most egregious examples of this kind of psychological bias. And so therefore, the confrontation of statements, oh heavens, I went the wrong way, of statements with empirical data, but we have to gather multiple sets of data, either from multiple people or multiple empirical signals, this then becomes the foundational concept of science, and that's where we begin to move forward. So it's how we can allow us to have the epistemological idea of truth arising from observation, because just an n equals 1 observation isn't good enough. And they figured that out back in the 1500s. Well, most sciences figured it out because we still have a problem with taxonomists. Okay, so, oh no, what did I do? How do I get back to the screen? Sorry about this, guys. I guess I do here, right? No. Oh, here. Okay. Yeah, we're there. Okay, now I'm going to tell you a story from my past. In fact, this guy here, Terry Fritz, is one of the reasons I am standing here in front of you today. One of the reasons I became an ecologist was because of the fascinating things he was doing in my home state of Iowa back in the 1980s. And he was a trained geologist, in fact, a paleontologist, and he did invertebrate paleontology, and he transferred his knowledge of invertebrate paleontology to invertebrate biology and started looking at the modern land snails of the upper Midwest, and he was the only person doing that in the 1980s. There was nobody else around. Oh, there we go. So, what did Terry do? Well, he's a geologist, and he figured out that a number of the rarest snails and rarest plants found in that part of the world were associated with a unique geological happenstance in which there were ice caves, buried ice caves that were where the entrances were covered over in loose rock rubble and that oozed out air just above freezing for the height of the summer. And what it did is it created small pockets of full glacial conditions in the modern interglacial landscape of Iowa. And sure enough, if you go to these places, what you find are, you know, some endangered plants and animals. There's the federally threatened northern wild monkshood. Here's the federally endangered Iowa Pleistocene snail. They're incredibly charismatic habitats. You have fog banks coming out of these fissure cracks in the rock and bathing entire hillsides in cold fog during the height of the summer. I mean, they're just amazing places. But there's a problem. They weren't really known until Terry came around. We didn't know how to geologically predict where they were. We knew very few sites, and almost none of them were protected. And yet these are incredibly fragile habitats, easily lost because steep, unconsolidated rock rubble, destroyed by pasturing, destroyed perhaps by even scientific research. And because only 2 species were protected as had conservation protection ratings, only sites that had those 2 species, one of those 2 species, could have money to be bought and protected, because that's the way the laws work in the US. So because there were only 2 species that had legal protection, we had very, very, very few sites that could in fact have money available to buy them and purchase them. So, this changed, however, when Terry came around, because he not only figured out how to find these habitats, but he started looking at other snails, and he then reported to the local government officials that there were 8 additional undescribed new species on these snails that were limited to these habitats found nowhere else in the world. And if you include those 8 additional species with the 2 that were already there, you got about 70 sites that could be prioritized for protection that had at least 2 of those species present. So it was a great thing, and everyone was excited. We got to protect algific talus slopes, which is what he called these things, cold-producing talus slopes. But then the problems start, and I'm part of the problem, because I was hired to be his field assistant for his last field year, and when I finally got my PhD and I got interested in land snails, I decided, you know, I should go back and take a look at these sites and gather some ecological data, because Terry didn't do ecology. And I collected lots of samples, I saw lots of shells, and the problem is that none of those shells look like his illustrations for these endemic species. Not a single one. His drawings didn't look like a single shell. Not a single shell out of the thousands I collected looked like any of his drawings. And not only that, but these different forms that existed, you could see continual gradation from one form to the next, to the next, to the next. And he said that was impossible. He said that didn't exist. So what do? Well, you decide that even though you're an ecologist, you're going to learn to do DNA sequence work, because how else are you going to test this? And that's what I did. I taught myself DNA sequence work. I did the analysis, and guess what? They were all one species. They didn't exist. In fact, they were all just shell forms of a different species called Vertigo arthuri that extends from Newfoundland to Alaska and all the way down the Rocky Mountains to Santa Fe, New Mexico. Got a huge range, and across that huge range, it has many, many different shell forms, and that's what his endemic species were. So, but here we have the same issue as Percival Lowell. Terry Fritz saw and believed. I worked with him. You know, he was not a bad guy. He really, truly believed these things existed. And so the question is, how can an intelligent scientist convince themselves that they're seeing something that simply doesn't exist? And the answers here are the same reasons. Terry received his PhD and his training in paleontology, and paleontologists can't generally go and get DNA and take a look at, look at the biology and the ecology of their creatures. They can look at the fossil record, which is basically just morphometrics. And so a species in a paleontological sense is not the same as a species to us biologists. It's kind of a convenient morphological, can that you throw things into. And so then in his mind, he connected various aspects of these shells to create some kind of Platonic ideal of 4 different Vertigo species that all differed in certain ways. And so, so he kind of refit the world to fit his notions. The sociocognitive factor was that, hey, 70 more sites got protected that wouldn't have been otherwise. And I hate to mention this, the other truth of the matter was that once he declared these endemic species to exist, grant money showed up. 10 years of it. In fact, I had a summer working for him that was based on these sort of fallacious arguments. And the cognitive dissonance comes in because he simply ignored the fact that you could see the gradation from one form to the next to the next. He just chose to ignore it. And then to sort of make fun of anyone who said they couldn't see it, that, well, they're untrained, they're just a dilettante, they don't know what they're doing. And that's how he dealt with people who tried to challenge his view of the world. So how did he get by with this? And the reason he got by with it is because we go back to the nomenclatural rules of zoology. And in the zoological code, you are not required to prove that your observations are correct. You just have to you just have to say how a species is different. You don't have to prove that your statement was correct. And so what the code is doing is it's going back to a pre-scientific, it's going back to that scholastic point of view, which is basing truth on what experts tell you is true. Terry Fritz says these things exist, and based on the fact that he's an expert, we believe they exist. And so what's interesting is that even 10 years after I've shown that these are not valid species, you take a look at the official endangered species list of the states of Iowa and Wisconsin, those taxa are still listed. And in fact, those are considered nomenclaturally valid names. Now we can argue whether they're biologically true, but in a nomenclatural sense, they are accepted. So the problem is that because the rules of how nomenclature and taxonomy is done is based on a pre-scientific epistemology, we allow species to be based on opinion. Somebody says there's a difference, here's the difference that I see, and we have to trust that that person is speaking accurately. And of course, it's an n=1 sample size, and we have no reason to expect that a single person's observation is going to overlap with anyone else's. So, because of that, the concern is that taxonomic categories might say as much about psychology of humans as they do about actual biology. So, this appeal to authority, how much of a problem is it in taxonomy? Well, it could be really bad. In the case of mollusks, people have done meta-analyses of 20 years of species descriptions across the mollusca have found that only 5% of newly described molluscan species even go to the point of measuring morphological variability within the species. And many less than that, probably 10 times less than that, actually try to test the null hypothesis that this new form that they're describing isn't just some form within a previously described entity. So the fact is, at least in mollusks, taxonomy is being done in the scholastic tradition, not the scientific tradition. So, and that's where the taxonomists will say, well, but we are being scientists because we're describing empirical differences between species. And the problem is that as long as it's being said by one person and there's no data to back it up, to prove that those observations are correct, we're ultimately still accepting on faith that the words of that taxonomic author are true. And that's no different than Thomas Aquinas saying 40 angels can dance on the head of a pin. Why? Well, because I say so. So how do we make taxonomy a modern science? I mean, that's really the issue here. How do we encourage the taxonomic community as a whole? And of course, the problem here isn't all taxonomists. It's some parts of the taxonomic community. And I'd say it's worse amongst invertebrates on average, but there's certainly trouble with other groups as well. I mean, there's a suggestion that up to 70% of amphibian species that have been recently described are in fact not valid species. So who knows? But how do we do this? Well, what we have to do is we have to bring empiricism. We have to bring modern epistemology, modern scientific epistemology to play. And so because that, we have a hope here. And the hope is that if we believe in biological species as evolutionary units, then what we're saying is that you're an evolutionary unit when you become independent, when you're taking your own independent evolutionary pathway. And when you do that, that independent evolutionary walk that you take is going to be influenced by different selection pressures, different random walks, different random events, different interactions. And so it is legitimate to expect that a valid species will show distinction from everything else across some consensus, across a multiple number of independent signals or quasi-independent signals. So in this case, we have a species from the western US called Pupilla hebes, and it was being misidentified and misclassified because it had many different shell shapes and sizes and amounts of teeth in the mouth. But one thing that held it all together was the same microstructure. And when we take a look at things showing that same microstructure, well, they all hang together well in separate, independent or quasi-independent, DNA signals, mitochondrial and nuclear. And they have, a very specific range that you can expect for it. So you have multiple channels of information that say Pupilla hebes is real. So now that approach that I'm just outlining, where we take a look at multiple signals, this is not something that would be unique to biology. In fact, our colleagues in astrophysics are doing the same exact thing right now. And they're calling it multi-messenger astronomy, where you take a look at something in space using multiple empirical signals. Multiple messengers, as they put it. And it can be visible light, It can be gamma rays, it can be cosmic rays, it can be neutrinos, it can be gravitational waves. And what the astronomers are figuring out is by looking at a single feature through multiple signals gives a much more complete and accurate idea of what's actually going on. And so I would argue that we should start doing multi-messenger taxonomy, where we take a look at some particular entity and we assess whether it is a valid species or not because it is distinguished, it is separate from other species in its mitochondrial DNA sequence, its nuclear DNA sequence. And I should point out with nuclear, it gets more complicated because we're really talking about different linkage groups could have different signals. So we could have multiple nuclear DNA signals from different linkage groups. They can have different sculpture on the outside of the shell. They can have different shell shapes. They can have different body colorations, ecology, reproductive anatomy, biogeography, et cetera, et cetera. And so it turns out, of all things, you can find a great summary of this process by reading Sherlock Holmes, and in The Disappearance of Lady Frances Carfax, Holmes tells Watson, when you follow separate chains of thought, Watson, you will find at some point, or you'll find some point of intersection which should approximate the truth. That is how you do multi-messenger taxonomy and how you take taxonomy from a scholastic tradition to a scientific tradition. Okay, so now let's put the rubber on the road and let's say, okay, what happens if we change our epistemological rules? If we go from the earlier idea to the modern idea of what is truth and what is knowledge, how much difference is there? And so what I'm going to do here is we're going to take a look at 3 different genera that we have done entire surveys of all traditionally claimed and understood taxa across the whole Arctic. And that includes 10 species of Euconulus, and Euconulus is this guy in the middle, about, let's see, 24 species of Pupilla, and 105 species of Vertigo. And what we're going to do is we're going to compare what the DNA and the multi-messenger approach tells us about what is a valid species to what was traditionally understood. And we're going to look at 3 forms of error. One is, okay, the entity was correct. It really is a biological species, but the taxonomist chose the wrong character to identify it by. A second thing is the species wasn't valid because it was just a biologically unique or segregate. It was just a part of another species that was already named. And sometimes the species was real but had been lumped incorrectly with something else. So we're going to look at those 3 different forms of error. And what it turns out, shockingly and disturbingly, and this was 3 groups of snails that were considered really well known, not controversial taxonomy whatsoever. There were no fights happening at taxonomic meetings about these groups. Less than half of them were both correct, were actually biologically valid and identified using the right features. Okay, over a quarter of them were being incorrectly diagnosed. They were using the wrong features to identify them, like we saw with Pupilla hebes. Another, just over 25%, were oversplit, where they were segregated out from another species when they shouldn't have been. And half of the species that were incorrectly diagnosed turned out to actually be valid species of their own, and they had been incorrectly lumped together. So it turns out that over half of the species that traditional taxonomy was using these groups were wrong. Okay, well, what's the impact of that on ecology? Because I'm not a taxonomist. I mean, I do taxonomy because I have to, because if I don't get the taxonomy right, I can't get the ecology right. So what's the impact on ecology? And this is work that just came out a couple of days ago in Ecography, so go look it up if you're interested. What we're going to do is we're going to take a look at the impact on ecological pattern at 3 different scales of analysis. One is the site scale, Second of all are kind of regional biogeographic scales, kind of large regions, and then biogeographic zones, really big, much bigger areas. We're going to have 9 different zones across the whole Arctic, 42 zones across Eurasia or North America. So, first of all, let's take a look at species richness. Were traditional taxonomic ideas misrepresenting richness at the site, regional, and biogeographic zone scales? And the answer is for sites, well, it depends where you are. In North America, about 10% of the sites were incorrect in their estimated richness, and of those, the great majority were over-reporting species. Okay, so the actual true species richness had to fall, was smaller than what was being reported. In Eurasia, it basically wasn't a problem. However, at the regional scale, 2/3 in both continents, 2/3 of the regional species pools were wrong, and in the great majority, too many species were being reported. And at the biogeographic zone scale, 100% of all the regions had too many species reported per region. So it turns out that, yes, this does impact estimates of biodiversity and richness of species pools, and the problem becomes bigger with the larger the spatial scale of analysis. How about the composition of individual records? We were looking at just the number of species. How about the species lists themselves? Well, it turns out just almost 50% of North American sites had incorrect species lists, and about 1/4 of Eurasian ones did. However, that increases to more than 90% for the biogeographic regions, and 100% of the biogeographic zones had incorrect species lists. So again, the problem gets bigger with the larger the spatial scale, observational scale. Now, turnover is when you take a look at 2 different areas and you say how much change was there in the species present. And so in North America, 12% of sites had altered turnover at less than 3,000 kilometer distances. And sites that were more than 4,000 kilometers apart, it was 50% of the site pairs had incorrect estimates of turnover. In Eurasia, it was much less, as 2% of pairs at less than 1,000 kilometer distances, but it increased to more than 25% at, again, more than 4,000 kilometer distances. And these were statistically significant. Now, it turns out that there is a bias to this, a predictable bias. And you see it both at the regional scale and the biogeographic zone scale, and that is the traditional estimates of turnover were too low at short scales. So this says that the change in similarity as we went from traditional taxonomic concepts to validated taxonomic concepts, empirically validated, that the actual similarity increased at short distances, but it decreased at longer distances. So in essence, at short scales, using traditional taxonomic concepts, we were underestimating turnover, but overestimating turnover at the larger scales. And so it turns out then that we were underestimating at the regional scale the actual rate of turnover. Actual turnover was higher than we thought it was based on these incorrect taxonomic concepts. And the exact same thing was true at the biogeographic zone scale. So again, the reasons for this is because it's the psychology of taxonomists. You come to believe that you know your local area really, really well. And if you see something that looks different, you're going to call it a new species. And that leads to oversplitting. So oversplitting predominates at small scales, but up until recently, there were few taxonomists who really worked at a global scale. And if they saw differences between organisms at large spatial scales, they'd probably just write it off declinal variation and say, oh, well, they must be the same thing, and there's intermediates in between. And so they actually overlumped at large spatial scales. And the process of Small scale over splitting and large scale over lumping causes us to bias our estimates of turnover so that we actually underestimate how much biological uniqueness there actually is in the world. Okay, so unfortunately, not only were half of the traditional concepts wrong, but that leads to overestimating site or local, local richness, with the problem getting worse the bigger the observational scale. Also, lots of error in our species lists, and also we underestimate turnover rates, and these problems get worse and worse and worse the bigger the scale is. So those of us who do macroecology, me, this gets to be really a big problem. So why did I decide to quit, like put on a lab coat and grab a pipetter 15 years ago? Because I didn't trust the data. I didn't trust the taxonomy I was being given, and the only way I could deal with it was to go do it myself because no one else was going to do it for me. And so it turns out it was a good choice I made back 15 years ago when I did this. And it took 15 years to realize, that I did the right thing. Now, I have a second part to this talk, which is the domination of authority is more than just accepting what an expert tells you about taxonomy. There's another aspect of this that is even more worrisome. And this idea came to me from, I used to work in Jim Brown's lab at the University of New Mexico. And Jim Brown, the father of macroecology, will tell you when asked, his biggest regret about how macroecology has developed is that macroecology makes so much use of mined data. In other words, data that you find, other people's data that you apply to your questions. And he is coming to the conclusion that this is probably a big problem. And it turns out that, it is a big problem, because if you just take other people's data sets and use them as if it was your own personal biases and errors are going to be multiplied with against those other people's biases and errors, because they're not going to be exactly the same thing. And at some point, if you have enough different data sets with enough different people involved in collecting it, you could have a lot of garbage produced. And so I would argue that uncritical use of databases like GenBank, Dryad, museum record databases, iNaturalist in particular, are going to lead, it's essentially a form of submitting to authority. You're saying that these data are accurate, and you're not going to ask the question, what's the level of inaccuracy in this data? Now, some people do, and in fact, when they've done that in some invertebrate groups in iNaturalist, it turns out you have a 60 to 70% misidentification rate of some insects. So, we already know that there's a lot of garbage out there. But the question is, can we identify impacts of collection bias and also labeling error from mined data? And what we're going to take a look at specifically here is museum record data. Now, the data we're going to look at is from Texas and the USA. I got a small grant when I was in New Mexico, my last years in New Mexico, to help the state of Texas redo their conservation prioritization lists for land snails. And so we decided to do two things. One is we were going to go through the two collections that have 95% of the world's Texas land snail holdings, which is the Perot Museum in Dallas and the University of Texas El Paso Museum in El Paso. And we were going to look at every single Texas land snail lot. We were going to make sure that the identification was correct. And then we were going to also mark down other sorts of potential errors in the labeling. And it turns out 150 species we encountered this way, total of 2,600 lots, 3,800 lots total between the two museums. And then I was also insistent that if we did this, we needed to also do ecological field sampling. And so at the same time, we collected about 100 sites across the entire state of Texas. We encountered roughly the same number of species, 130 about. 1,050 total species occurrences, 52,000 individuals. And I will say that one of the things that really differs between the two data sets is that we really avoided sampling anthropogenic habitats, whereas not all of the museum data lots were collected from pristine natural habitats. Okay. So what I thought we should do is compare these two data sets. We have a view of Texas land snails based on museum records, and then we have the stuff that we went out and collected by hand. How do they differ? How are they the same? And it turns out, well, okay, there are collection biases. And the reason is people who are working in museums and collecting biological material are not ecologists. They're collectors, and they're trying to answer taxonomic questions. And, you know, they're often really excited about a certain group of organisms. And so they're going to collect in places where their favorite organisms are, or they're going to collect in places that are known to be really diverse because they're thrilled by diversity. And so, and they're not necessarily going to use standardized methods that are going to actually sample the entire fauna. So one of the things you discover right away is that museum collections underrepresent. So the dashed lines here or I guess the lines are ecological samples. And it turns out museum records underreport small snails, so snails less than 6 millimeters in diameter, but overreport bigger snails, things bigger than 6 millimeters. And that's because it's easier to see the big snails, and you can pick them up, and it's far more fun than, you know, getting yourself dirty collecting leaf litter and then going back to the lab and having to sort through leaf litter to pull out tiny snails, you know, on a wet brush. I mean, that's not as much fun. And so it turns out the museum collections are biased in size of large species. There's another bias that was really important that fell out, and that is where the collections were being made. It turns out that collections in museums are not a random sample or even a representative sample across the state of Texas. In fact, they're more likely to be made in counties or areas that have more people. So they're going to go to places where their family or friends are and collect near to where family and friends or themselves are. So higher human population density. The larger the county, the more you know, likely it was going to be collected. Okay, fine. But also that there was a statistically significant effect of the presence of some iconic landscape feature, like a U.S. national park. So if there was a national park present, like Big Bend National Park or Guadalupe Mountains National Park, way more collections than there should be. And also, there was a significant negative correlation of collection numbers with distance from the museum. So the collectors are more likely to collect near to the museum and less likely a long way away. And so because of that, the collections that are made in museums are not a representative sample of what's going on across the state of Texas. And another thing, some people are wanting to use museum records, the abundance or the number of museum records of a certain species to tell us or give an idea of how abundant that species is in the landscape. Well, that doesn't work either. Because if you're a museum collector, you are less likely to collect some common species that you see all the time, and more likely to collect something that's rare because it's exciting and fun. And so like in my college herbarium, Taraxacum, dandelions in English, had fewer records in our herbarium than Cypripedium orchids, lady slipper orchids, which are, you know, many orders of magnitude more rare. And the reason is because if you find a lady slipper orchid, you want to document you saw it, you're really excited, it's really pretty. And a dandelion, yeah, who cares? And so that's turns out to be going on for these land snails as well. And so if you do a Spearman rank correlation of all species, abundances or number of occurrences of a species in the ecological samples and in the museum samples, you get a significant correlation between the real world of the museum collections only if you include the 5 most common species. And after that, there's no significant correlation. And so it turns out you can only estimate abundance for museum material only for the very most common species. And for the bulk of the taxa, they're not reflective one to the other. Okay. Well, there's also another whole issue. So we're talking about collection biases, the fact that people who are collecting for museums are not necessarily trying to get us an accurate representative snapshot of the whole world. There's another issue, and that is, did they identify the material correctly? And this is one of the things that I found most sobering, because these 2 collections in Texas had been put together by the experts, the Texas land snail experts of their day. So I expected misidentification errors to not be very big at all. And in fact, it turns out in both collections, those guys misidentified 20% of the material in their collection. And it turns out that there was a significant relationship to the shell size with, in fact, the sort of intermediate size things having the highest rates of misidentification. So the smallest stuff, well, they were more accurate because they didn't collect much of it, and when they did, there were very few shells, and there was, they could usually peg it right. But in the middle zone, there was a lot more things where they could make errors. They could be wrong in a lot of different ways, and they were. And it turns out this had a significant impact on the observed body size distribution. That in fact, the real world distribution is a really nice bell curve. It's much more uniform with the misidentified material. And lastly, there's just amazingly crazy labeling errors. So at UTEP, there's a place called Hutchinson County. It's up in the northern part of Texas, out in the flat plains. It's the last place in the world you'd expect anything interesting, because it's basically just oil wells and wheat fields. But lo and behold, there were 24 lots at the UTEP Museum that were collected on June 19th, 1971, it said in the labels, from along the road to Adobe Wells or northeast of Adobe Wells Monument. Now we got to realize there was a problem here because you go and look for Adobe Wells and there is no such place. There is, however, a place called Adobe Walls. And then these species, these 24 lots that were supposedly collected at this place that doesn't exist in Hutchinson County, turns out to include 11 species that either were previously unreported in Texas, or if they were reported, were only known from the tops of the highest mountain peaks. And well, that's what Adobe Walls looks like. There's no mountain peak there. But it turns out that when you went into that collection and you said, well, was there anything collected on June 18th or June 17th? What you discovered is that the collectors had been in northern New Mexico in the Sangre de Cristo Mountains, where every single one of those species that they reported from Adobe Walls, or Adobe Wells, I should say, is common. And so it's pretty clear that at some point, they transcribed their collection materials incorrectly with their field notebooks, and it got reported as being in the plains, the hot plains, low plains of Texas, when in fact they were mountain species from New Mexico. And so you kind of laugh and say, oh, well, those things happen, none of us are perfect. And that's true, none of us are perfect. But here's the problem: when we're looking at rarity, it's the outlying events that drive rarity, right? So it's these kinds of errors in labeling that have an oversized impact on our ability to do conservation. If we took those 24 collections on face value and reported them from Hutchinson County, Texas, we would have misanalyzed and misreported the conservation status for fully 5% of the entire Texas land snail fauna. So these single odd mistakes in the end, because we're studying rarity, not the average commonness things, but the rarest things, these rare events end up impacting our observations of rarity in a great way. So, the naive use of museum databases to tell us about ecology means that we underreport the smallest organisms. It means we bias our collection information in favor of the areas with more people, or that are closer to the museum, or that happen to have some iconic destination. It also means that we're going to over-report rare things, and we're going to mean that we can't actually accurately tell, except for the few most common species. We're not going to have an accurate reflection of abundance in the real world based on what's in the museum collection. It means that 20% of the lots are going to be misidentified. And I said, well, you might say, oh, well, that's just those two museums. Well, it was two different people, and they both were wrong 20% of the time. And it turns out that that 20% number comes up over and over and over again. So for instance, there was an analysis of the identification accuracy of bird skins in the Scholasticism Museum in Slovakia. Guess what rate the bird skins were misidentified? 20%. I'm seeing that 20% factor come up over and over and over again for humans. And I actually am getting to the point where I think all of us are wrong 1 time out of 5. It's just the way we are. It's part of being, you know, a human. We're wrong. We're wrong 20% of the time. So, but those identifications lead to a flattening of the apparent body size distribution. And also, if you're doing conservation activities looking for rare things, these rare transcription errors can actually have an outsized impact. So, this is the end of the story then, and you guys can have at me and tell me why I'm wrong. I think we need to be extremely cautious when we as scientists in the era of big databases and global access of information, when we do not own our own data, when we submit to the authority of other people's databases. And it's just true in a couple of different ways. In taxonomy, it means accepting what some taxonomic expert tells you and just say, okay, sure, the expert in the world tells me this, I'll accept it. Well, that's Thomas Aquinas telling you there are 40 angels dancing on the head of a pin. Okay, we have to validate, and we have to document that those entities are real. And we need to start encouraging the taxonomic community to move away from that scholastic epistemology into modern scientific epistemology, where we validate our ideas based on multiple data streams and multiple observers. And the thing is, if we don't do that, we're going to impact in a negative way the way we are documenting biodiversity in the world. In fact, the most important thing to me is that we underreport the uniqueness of regional faunas. So that's something that I think is really important. Also, if we are mining data sets and we just naively say, oh, well, there's some good data, I'll just drop it in my data set. I mean, I think, to my mind, I've wanted to do this, and I have done it. I've mined data, and it's kind of like eating bromburki, right? Oh, you have one, oh, that was really good. Oh, I want some more. And, it's addictive. It's addictive to have free data handed to you. But there's a real problem to it, is that if you're taking free data, you're also taking along with it the biases and errors and perceptual psychological issues of the people who assembled those data sets. And I actually think these easy access now to all this collected data from various observers, like the fact now that many journals, make you want you to put all your data, into these accessible databases. It's just feeding that addiction. And I think what we need to do, we need to, realize it's an addiction, and we need to own the data by going through that data, and before we put it in and use it, we have to check it for errors and biases and try to make sure it's accurate. And therefore, as in Nullius and Verba, what is it that they were trying to accomplish in 1600 when they 1660 when they came up with that phrase? They wanted to withstand the domination or the dominance of authority. And unfortunately, 400 years later, well, we're kind of back to the dominance of authority because it's so easy now to get other people's ideas and other people's ideas and not own them ourselves by checking it and making sure that we agree with them. And so my plea to you all is that as you do ecological research, trust no one. You know, make them prove to you that their data is real. Check it, make it your own by validating that it's good. Don't just grab data off the internet and then drop it in. I mean, it could be the, you know, God knows these days where that data came from. It could be, you know, the Russian Secret Service created it, for all I know. So trust no one. Use our tools that were invented at the beginning of the Renaissance, our empirical observational tools, to vet and make sure and prove to ourselves the data is real before we go ahead and analyze it. Lots of people think, and I'm glad I kept your interest for this weird talk for all this time. You can have at me now. Tell me how I'm wrong. Well, yeah. Okay. So one of the things that we know by looking at what's called taxonomic load, which is, or synonymy load, which is, you know, overlumping, is that overlumping rates appear to be higher the more charismatic the organism is. So the more visible and the more people there are doing taxonomic work on that particular organism, the higher the rates of taxonomy load. And so in land snails, there are over 140 subspecies named of the Florida tree snail, and it's all just different color morphs. And it turns out once we got DNA data, guess what? They were all the same thing. So I'm not so certain. In fact, I would say in the land snails in North America, I think it's a totally unanswered question about how many of the supposed single mountain endemics. Many of our 1,200 land snails in North America are supposedly restricted to single mountains in the West. And yet, when I look at the one that's from the Sangre de Cristos and compare it to the one from the Huachucas in Arizona, boy, they look the same. Now, until we get the DNA, we won't know. So in one hand, I agree with you. On the other hand, there's also suggestions that, in fact, these taxonomic issues become worse the more charismatic. In fact, with plants, there's been work done to show that orchids are named at a much higher rate than sedges, even though there's no inherent reason to necessarily believe that, that there's actually a difference there. So anyhow, yeah.
Yeah, I agree also with this. I work in entomology.
Mm-hmm.
It's true that in groups which are large, colorful,
Yep. Yep. Absolutely. Yep. Oh yeah. Yep.
So I don't think taxonomy is very special among science. There are good taxonomies and there are
Yep.
bad taxonomies like
Oh,
and
yeah. I'm totally agree with you, and so my criticism of taxonomy isn't the individual players. It's the fact that the code hasn't updated itself since the 1500s. And the code should not allow anybody to say, I think this is a new species because I think it's different in this way. And that's allowed. And in fact, the code doesn't even require that you publish it in a peer-reviewed journal. It has to just be accessible. And of course, talking about angels dancing on the head of a pin, you should see the arguments, at the ICZN about what that means. So I guess I would say my criticism is that if by some miracle you could change the rules of nomenclature to update them to the 21st century, a lot of this would sort itself out pretty quickly. And there's a lot of us doing good taxonomy, and I wish we didn't have to, be correcting all the sloppy, lazy, citizen science people. I mean, we've got a terrible thing. Well, actually, it's a land snail guy, but he's doing it with beetles. We've got a colleague who left Naturalis in Leiden to start his own company, and he has people paying him to go out on collecting trips with him, and then they get to name species. So he's doing sort of self-funded citizen science by total amateurs. And it's an interesting business model, but what is it doing to science? I have no idea. But that's where...
There was a talk given at Zoological Days by the young guy from Prague. We are talking about the same guy,
I mean, I love him. He's a great guy. But I mean, it's like... that business model to fund his life. I went through a bad stretch, and I was barely able to make it, but I never did anything like that because I believe in science, so I don't know what to say. So I agree with you. If you hear this as me saying all taxonomists are bad, absolutely not. There's a lot of us doing 21st century science, and people who are doing a microbial taxonomy are way ahead of us because it's all being driven by this multi-messenger taxonomy approach. The problem gets to be things where we can still be medieval. We're still allowing medieval thinking to take place, and some people take advantage of that. I don't know. What can I say? But I fully agree with you, and I'm sorry if it came off as a broad brushstroke against all taxonomists. It isn't.
Thank you, Jeff. But do you really think that the problem is in the code of zoological nomenclature? And, I would say there are two things. One is taxonomy, and the other is nomenclature. And,
Yep.
The code is not prescribing how taxonomy should be done.
Right.
The code is just a tool how the names should be created. And if someone is doing bad taxonomy, then code will not help.
Yeah.
So first people have to be sure that this and this is a species or a taxon.
Yeah.
And then they can give a name, but I don't think that the code should prescribe how taxonomy should be done.
Okay. Well, that's fair. I mean, I think what we say in our ecography paper that just came out is that ecologists need to not treat taxonomy as a black box. We actually need to take a look and say, this naming system that we're using for this study, how was it being supported? And that when we have a choice, if we can choose naming systems that are based on a more empirical hopefully multi-messenger approach, we should trust those over other naming systems. So I guess part of it is that because I'm not a taxonomist, I'm an ecologist. And so really what I think that's the most important thing to me is that we need to quit just saying, well, we'll accept whatever the taxonomists tell us because, you know, for whatever reason. I think we need to actually say, what's your proof? You know, why do you say that? And then choose the system that's most supported, I guess.
I think that sadly, in the community there is always, quite often, a toxic taxonomy. Everybody knows it, but you can do hardly anything about it. If he's publishing rubbish, you have to clean it, you have to accept it. Even everybody knows it's just a nonsense. So maybe this is the room to at least, you know, push the thing forward. But otherwise, find some rules that you have to fulfill if you want to describe in your speech.
There are also ecologists who produce a lot of rubbish.
Yeah,
Well,
in the ecology, you can just say, okay, this paper rubbish, and it's done. But with the name, you have to really did some revision. It's not about just the saying or showing that's the bad. You have to really get more. Otherwise, it's still valid.
You cannot ignore it.
Well, you can ignore it, but the code makes it real. That's the thing. In other
The code doesn't make real the taxonomy. It makes real the name.
yeah.
I think the two get confounded.
Exist without the name.
So I think the two get confounded, and that's the problem, is that for some people, the name is the taxon. And even though we can understand, okay, that's really not reality, for some people at least it is. And that's where it gets confused. But I don't know what to say other than, I guess, to just admit that there could be a problem. And, you know, I think the way forward is like Galileo did, and let's collect our own data and let's see what's going on. You know, that's what I would say, if we can.
I should not be defending taxonomy. I'm as much a taxonomist as you are. So but you said that we have to take what the taxonomists tell us at face value.
Mm-hmm.
But actually, I mean, there is the type material. And that's a very important thing. So basically, you can always, I mean, the idea is that you can always go back to the type and prove that what he or she described is absolutely rubbish because it doesn't look that way.
Right.
It's a bad drawing.
Oh, yep.
Okay, we have to say that many type material have been lost over time, or maybe from the start and not admitted. You know, I mean, that there were times when for certain groups it was not a real requirement, or it was not enforced that there has to be
Yep.
type material. For some groups, it's very difficult to preserve the organism in a way
Yep.
that you can actually see
Oh, yeah.
something. So that's really a problem. But you have this type. So that's because in a genetic study or ecological study, basically when you make up your data and you do it in a wise way, it's quite difficult to prove. And you know.
Yeah. No.
But here you have the type. I mean, so I would be a bit careful to.
And so what we do is often I try to collect material for genetic work from as close to the type locality as I can. And that becomes the gold standard for what DNA should be associated with this nomenclatural name. And so it turns out that, you know, the finding type localities or near type localities for things becomes hugely important. And you know, the other problem is, as Michael alluded to, is that I would say it takes 10 times more work to sink something than it did to erect it. And so I wish that it was harder to erect things, because it's so much damn work to look at the types and to try to go to the type locality and, you know, and figure out where it is based on historical records, and then go there in person and get material for DNA analysis. And you know, I mean, you got lucky, you know, in Pyramidula, there's something called Sexatulus turns out there was the type material was in Vienna, and actually we were able to get DNA out of this big lot. There was hundreds of shells, and so we could sacrifice one of the shells and get DNA from it and show that in fact Saxatulus is this. But it's a huge amount of work, and I just wish that the threshold for naming something was a little bit harder. I mean, that's
impossible, like our problem with one of those.
Oh.
Just, lost the types. The curator lost the type, so it's impossible to get the type. So, well, but they exist.
I mean, they're in her house somewhere in Poland. They're in her house somewhere, probably in back of a vodka bottle. The problem is that we're discovering that there's a diversification cluster in the Himalayas for this group of snails, and she named 2 things, but we don't know if those are the same things that we've been finding or whether they're different things, part of the group. We have no idea until we look at her type material. And we've been asking now for 2 years, and it hasn't shown up. So we don't know what's going to happen. But I do just wish that naming was a little bit harder, which goes back to the code. Because I think if the code said description must include these sorts of empirical tests, and of course it'll never happen because one of the things that's really unique about taxonomy is it's very pluralistic, and as long as you're able to follow the rules, anybody of any training can name life, which is kind of cool. But it's also a huge issue because anyone can name life. And maybe those of us who've trained in it, should be the ones who, are given the keys to the car. I don't know. I don't know what to say. Yeah. Yeah. Mm-hmm. Yep. Yeah. Yep. Sure. So so my question is that such a problem that we're willing to have more than half of the names that get erected be wrong? And at what point is that noise an impediment in and of itself? And I'll give you the example. Terry Frest, okay? His endemic snails, none of which are real, not a single one of them, influenced conservation strategy in the upper Midwest for 20 years. And we protected sites, well, in a way that we protected more algific talus slopes, but there were actually endangered snails in endangered habitats like fins that didn't get protected. And so there were endemic species present in that landscape that were endangered that got left out because we had garbage names. And so there's a cost as well to lowering the threshold, to make naming easier, to name all those unnamed amount of life. I'm getting to my history with Terry Frest and the algific talus slopes is I'd rather be slower and do it right. It's like if you were a carpenter, and I don't know if there's a Czech saying that's equivalent, but there's a saying where you measure twice, cut once. Okay? Okay. I think biodiversity protection is so important, we should measure twice, cut once. And if that means we're a little bit slower in naming things to make sure we're naming good stuff, my preference is that we do that myself. But I certainly have had people say that to me when I have rejected, when I've sent manuscripts back to somebody and say, well, you didn't test this. I'd like to see a support value or a p-value for this entity that you're trying to name. And they get upset with me and they say, well, but most of the world's life is undescribed, and if we don't name it, we can't protect it, and you're hurting the world. And I'm saying, but you could be hurting the world if it's all a figment of your imagination. Prove to me it isn't. And usually we just quit talking to each other. I mean, they're upset with me because I quit them publishing their name, and I just go roll my eyes and say, oh God, what does it take to become a 21st century scientist? So I don't know what to say. I think there's a downside to that as well, of just opening up the spigot and naming everything we can see.
I kind of agree, that some even like established taxonomy, Nullius have trouble to see the name or the new taxon as a hypothesis. The hypothesis that you are going to test. For them, it's a fact. They saw it something to be tested, so then they would probably have less trouble to understand if I say, okay, to claiming that these two species differ in number of ribs on a shell, show me a simple box plot. There is a significant difference. Just the material you have, no matter that could be just scientific plasticity, but at least do this for me, because based on the table I saw there is a huge overlap. And they claim they refuse to do it because some reason. So I mean, it's already this fact is showing, okay, maybe those are good species, but definitely not based on the characters you are raising.
So let me, I've got a story from the plant world. And at the University of Michigan, back when I was an undergraduate and graduate student, there was somebody by the name of Herb Wagner, and he got to be known as the world's expert in Botricium, small fern genus. And these are the Botricium lunaria, I don't know what the common name, moonworts. So anyhow, the problem was he started naming all these, he had dozens and dozens of new moonwort species from North America, and we couldn't make sense of it. I mean, it just didn't make any sense at all. And so the first university that I taught at, University of Wisconsin-Green Bay, Gary Fulis, the taxonomy curator, decided to do this really evil experiment. So he sent Herb Wagner 20 sheets of Botricium and got him to annotate them, okay? Sent them back. Somehow Gary was able to get the annotations off the sheets. Somehow he anonymized them. He sent them back to Herb 2 years later. And the names changed. I mean, it wasn't that he named new stuff. It's like he was putting things, like his own classification accuracy within his own head wasn't accurate. He was, and so at that point, I think if you're splitting to the point where you can't even keep track of it, maybe that's a sign that it's an issue. And I don't know genetically that we've ever worked out whether, how many of Herb Wagner's Botricium species are real. I mean, I don't know that we've done that yet. But anyhow, yeah. Sure. Oh, you don't. I'm wrong 20% of the time.
20%.
I'm wrong 20%. No, so no. There is except when I'm using data. If I am using multiple streams of empirical data to come up with my rules of how do you identify these things, then the error rates fall. So for instance, in the genus Euconulus, after we went through and did our DNA work, I went back through my collection and re-identified things after I had, confirmed what the actual diagnostic characters were. And I was wrong, but 5% of the time, 20% to 5. Now, of course, that's my eyes, and what would somebody else, if I had Michael go through it and do that, it would, he would have gotten probably more errors than me. Might have gone up to 10%, but still, I still say that I believe in the empirical method, and I believe that it can get us past our personal psychology because of how successful that epistemological revolution was. That you look at how much advancement we had during the Middle Ages, and we had Galileo and Da Vinci and Bacon and these other people, Copernicus, over north of Prague, changing the epistemological rules, and look at how far we've come since 1500. And I think that it's a method that works. I think we've got good evidence that even in spite of the fact that we are all constrained by our own biases and errors, as soon as we replicate and we also use machines to see the world for us so that we're not seeing it only with our own eyes, and we have multiple channels of information and we choose truth based on when, as Sherlock Holmes says, there are multiple channels of information and where they intersect, the truth probably lies there. If we do that, I think we're less likely to be wrong. So I'm still making errors. I mean, you know, because I'm human. No, never. We can't be 100, but we can go from being wrong 1 time out of 5 to 1 time out of 100. We can change that, you know, so we can, huh? Why? Because, why we should is because if we don't, we are going to have messy data and wrong conclusions. So, you know, if we used the traditional taxonomy to map climate envelopes for these snails, you would be off by, you know, either being 3 times too big or 3 times too small for envelopes. And we know that because we've gone through the data and calculated it. So the point is that the more you can limit that error, and if you can go from being wrong 1 out of 5 times to 1 out of 20 to 1 out of 100, each of those steps means that the data that you're analyzing and interpreting, the more accurate your data becomes, the stronger your analytical outcomes. So those climate models we came up with are amazingly, I can't tell you, it's eerie how well they work. I mean, they're even picking up from using regional scale climate data, it's picking up what species live in certain microhabitats. I don't know. I generally don't believe models that they're that accurate, but I'm now thinking the problem with modeling isn't the models, it's the crappy data we're feeding into it.
Or a model, we have a good one.
Yeah. Right. So if you create a model based on data that's only wrong 1 out of 20 times versus 1 out of 5, it actually makes a huge difference down the road in how strong that model is and what you can say about it and what it tells you about the world. So I think that's probably the next paper I want to write in this sequence is I want to start working with some mathematicians and try to figure out if errors between observers are multiplicative or additive, and how much error do you need to add in before you can't interpret the results anymore. And I think we can get at that. I mean, I think with the right mathematician, but what I'm concerned about is that errors are multiplicative and it doesn't take many different 20% error rates before the outcome is total garbage. I'm really worried about that.
So in the talk, you mentioned oversplitting and overlapping.
Yes.
And now the whole discussion was about how to reduce or stop oversplitting.
Oh.
There was nothing about overlapping.
Oh, yeah.
Yeah. And I think because there were like proposals that we should reduce standard for describing new species by changing the code or I don't know, which would reduce oversplitting, but it would increase overlapping because I think there will always be errors. Yeah. And oversplitting and overlapping are kind of trade-off. So
Yeah, type 1, type 2, yeah.
the more overlapping and vice versa.
Well, okay. Yeah, I get it. And the only thing I can say is that if you structure your sampling correctly, if you get your DNA from the full known ecological, geographic, and morphometric range that a species has, you're going to see those overlumped things that are in there. That's how we found them, you know. And so the only new species we've run into recently in these groups that we've worked with have actually are things that are actually would be considered just new species. They're not things that have ever been lumped with anything. It's just like you see that and you say, oh, I've never seen anything like that before. And so I think that you're absolutely right that overlumping is also a huge issue, and it happens at roughly half the rate of oversplitting. And so, and how you deal with it is that when you do those empirical analyses, the multi-messenger empirical analysis, you need to make sure you're doing it across the known environmental, geographic, morphometric range of a species so that you can be picking up on those things.
So now you require to do really a huge work.
Oh yeah. Oh yeah.
Collect everything over the whole
Yep.
range.
Yep.
So no one will now travel to Russia for
Well, that's well.
we will not have any possibility to do such studies over the Arctic region, for example. So we will not describe new species, even though they may well exist and they may be in need of conservation.
Well,
So this is again the
well, except we've got material from before Russia went evil. So we've got,
When was that?
okay, okay, they were more drunk and less evil. Okay, so when there was higher alcoholism. But the thing is that we have archived in museums, we have tissue samples. So we can, and the thing is that I guess what I'm trying to say is that I don't think taxonomy should be easy. I guess I really fundamentally believe it should be hard, because to really validate that something is biologically real isn't easy. And I think we have made taxonomy in a way to be easy to be able to pump out as many names as we can. And I think that may be hurting us down the road. It certainly has made our lives more difficult as we've had to deal with spurious names that we've had to sync, and that's, but I guess it's possible to get, I mean, yeah, we can't go out and do field collections, but we can find other sources of data. And I think we can, and we should, when we can. And if not, we need to just then put an asterisk by say, here is our analysis, but in the discussion section, say, here are potential sources of error, and we should be cautious about the following, about over-interpreting these results. And so I'd say, if nothing else, we as ecologists need to be upfront about, one of the things in epistemology is how far can you go before it's no longer knowledge? And I think we need to start thinking about that. How far can we interpret data before it's no longer knowledge, but it's belief? And so you're right. I mean, I don't know. The real world is messy, so what can we do?
I have a comment. One of the probably, for me, the coolest discovery from that study is that these two processes, oversplitting and overlapping, is scale dependent. So you can oversplit a smaller scale one on a bigger scale. So it's not like it's going against each other. It's operating on different scales. Might be different in different organisms, different regions, but mostly overlapping is if you have a good flora or fauna in one region, you travel to another one, or if you travel, you see, but people that are working in that region and having literature from another region, so they tend to associate what they are seeing with, because they are not sure, they never have an access to the material, they are not sure if they are cautious, if what they are seeing, these differences, just because most differences you can understood correctly just from the drawings, from the text. You have to see the material. So that's my point. And and also the problem is not that mostly that we don't have the material. Material is here, but it's a lot of work. You want to do some ecological analysis and first spend 20 years by correcting taxonomy is nonsense.
I mean, and getting funded, I mean, You can, I mean, you know, it's impossible. Okay, I mean, I'm going to be putting together a GACR proposal, and we're going to be trying to look at, you know, how stability of global refuges is impacting modern biodiversity. And okay, great, but you know, if I got to write my own proposal that I really want to do, I really want to go and travel the southern hemisphere and collect vertigo, because I know now that vertigo is not a northern hemisphere genus. It extends all the way down into Australia, and that oh no, we know that. I know I have a partial DNA sequence that I know is vertigo, and we know it's from South Africa, but what I want to do is I want to figure out what the actual, I'd love to just go to Tahiti and then go to, you know, New Zealand, and I want to go to the southern part of Japan, and I want to go to the Himalayas, and I want to go, you know, and there's no, oh, I know, it's just terrible. Hawaii, you know, I want to do this because I think that if we want to understand the evolution of this genus of snails, which is one of the most diverse genera in land snails, it's always been assumed to be northern hemisphere, and in fact, it's both hemispheres, and it's probably more diverse in the south, based on the tiny glimpse that we have. But you know, there's no way I'm ever going to get JCR to fund that, and just no way, because it's a small issue. It's not seen to be anything that's going to be global, that's going to help us live in the world, and so you know, I desperately want to do it. I'll never get funded to do it. You know. The best I can do is, you know, plant seeds of interest amongst the people of the southern hemisphere and hope they collect stuff and, you know, look at it. Because I'm never going to get a funding agency to send me to, you know, Tahiti and Hawaii and Easter Island and, you know, and the Guianas and, you know, Cameroon and, you know, and. It's never going to happen. Yeah.
I mean, we all scientists, we should strive to deliver good work.
Yeah.
I mean, no discussion about that, I hope.
Yeah.
But I mean, you've heard the term taxonomic impediment, and now you want to make it more difficult. But taxonomists are actually dying out. And why are they dying out, amongst others? Because nobody is willing to pay them, you know, to do this more complicated stuff. So I'm not sure, you know, what's the incentive for young person, for young biology to become a taxonomist?
But you know what? I think we're selling ourselves short, because by allowing taxonomy to operate on the rules of scholasticism, I think it makes it really hard. It means that the aggressive, that a certain personality type is more apt to be successful. And if we made it data-driven, I think it actually opens the door to more people who would be interested in doing taxonomy, myself. Now, of course, the current people who have chosen that field, because it's the one place you can still be a scientist, and I'll put that in quotes, and not have to really collect any data, numbers, you don't have to be empirical, you know, they're going to be really upset with me. But you know what? My concern with taxonomic impediment is if we get rid of the impediment by creating garbage data, have we done anything? And at this stage, I am learning that high quality data is everything. And if you have poor quality data, there's very little you can do with it. And so I would say that our way to deal with biodiversity issues actually is to make sure the data we get is better, which means, I mean, I'm one of the few scientists in the world who is really worried about citizen science. If it's so simple that anybody can go out and be a scientist, then why the hell did I just spend 30 years of my life studying to be a scientist? And I went through hell and back to stay in science. And if it's as simple as being a hobby, I think it sells us so short. And it also creates reams and reams of bad data. And bad data leads to bad conclusions. So, okay.
If I remember well, just talk about the mollusk data from different regions of the world. If I remember well, the data from Eurasia were much better in quality than those from North America.
Mm-hmm.
taxonomists in different countries, different schools, more discussion, more questioning of
Okay.
while in the US maybe it was 2, 3, 4 guys who did it all over the country?
That's a really interesting question. And again, it's sociology. Up until about 1950, the US, North America was way ahead of Europe as far as malacology, and they were at the cutting edge of everything. And then the 2 or 3 people who were inclusive and scientific, Henry Pillsbury, Clapp, Baker, passed away. And who replaced them were a few, that just by random chance, they were difficult people, shall we say. People who wanted to have become the Aristotle, their own Aristotle, okay? And so what you had then was 3 or 4 difficult personalities who took over and poisoned the well. And in fact, I was the first person to become involved in doing land snail work, basically in North America, that wasn't in that group of people. But they were fighting, they'd bicker with each other. Terry Frest was really a difficult personality. He hated everyone else who did land snail work in North America. He belittled them and made fun of them, and they belittled him in return until he died, and then he was revered, which I think is an interesting, when they're alive, you can make fun of them. But they scared off everybody, and I know people, Kostov Roy, who does macroecology work on nearshore marine organisms, wanted to get involved in land snails early on when he graduated from the University of Chicago out of the Dave Jablonski lab, and was absolutely prevented from doing it because the grand old man of California malacology wouldn't permit anyone else into his terrain. And so he was forced out. And so there were people who wanted to do it. And so what happened in North America is we were way ahead of Europe. And then by happenstance, these liberal minds died. They were replaced by these difficult, unpleasant personalities who essentially, through aggressive competition, chased out any potential competitors for the next 50 years. And it took somebody like me who refuses to be chased away. I'm a difficult personality in my own way. And I was just like, screw you guys. You're not going to publish my papers. I'll send them to Europe and they'll publish them over there. And that's what I had to do. So I sent my stuff to European journals initially because they would not allow my stuff to get published. Until eventually I developed enough they couldn't sit on me anymore. And by that point, they were already dying. And we had our own cadre of young researchers coming up. But, I was kind of at the front of the charge, getting most of the bullets. So I think part of the problem was in North America. And then why is North America had such a high rate of error? Partly Terry Frest. 40% of the over-reporting of species richness is just due to his taxonomic exuberance. But it was more than that. There was a few groups like Pupilla where Henry Pillsbury had just chosen the wrong characters to identify snails with. And so he didn't have any way to know what was plastic and what was robust, and he chose badly, as I guess to quote the 3rd Indiana Jones movie. He chose badly. And so it turned out that in Europe, probably because there's been more interaction, people were making better choices as to identification features. But North America went through this dark ages in malacology, and it just reminds me of how important it is to have an open door in science and let everybody in. And it's okay to have people challenge your ideas, because those old guys didn't ever want to be challenged, and it poisoned that field for a long time. Uh-huh. Oh. Yep. Oh yeah. Oh yeah. Yep. Oh yep. Oh yep. So it's a huge problem that, okay, there's something called the Mehe itch. The Mehe itch is when you get excited about describing new species and you just start describing new species because you enjoy it. The Mehe itch can happen with bad data, which is what you're talking about. So we've got a real problem, and not just in land snails, as you say, in many different groups, that people will take one channel of DNA data and use it to then declare species level patterns, but they're using n equals 1 data streams. And that's why it's so important we do multi-messenger, because in fact, there's a paper that Mikhail and I and Vercha, everyone knows Vercha, that we published about when is a cryptic species not a cryptic species, because we took this genus that's found across the entire whole Arctic. We got DNA from across the entire whole Arctic, and oh my goodness, it fell out into 4 different groups, 4 different highly supported clades, and there were 4 clades in the same topological position in nuclear as mitochondrial. And we're like, oh, no, these are cryptic species. And then we take a close look at the data, and it turns out that you could be in clade 1 in nuclear and clade 3 in mitochondrial, and they were all switching back and forth. And then there was problems where we could find where there was heterozygosity, and the 2 peaks that we got were usually from one clade or the other. So we could see there was hybridization occurring, and then we could show that there was no morphological differences in the shells. And the whole point of our paper was that simply showing that there are, highly supported clades does not mean that it's biologically real. And so that's a real problem. And I know, I'm one of the people I work with in the US does a lot of work with snakes, and there was some person in the Southwest who wanted to split a common snake into 4 different taxa based on only mitochondrial DNA. And I was telling Charles, oh my God, no. I mean, he's got to, get multiple channels. And then one of the insidious ways that you can take multiple channels and turn them into one is by concatenation. If you concatenate mitochondrial DNA with some nuclear DNA that evolves very slowly, well, if 95% of your variability is coming from mitochondria, and you just add in concatenate in the nuclear, guess what? The nuclear gets swamped out. And so even though they're saying in the paper, well, we looked at both nuclear and mitochondrial, they really didn't, because they turned multiple data streams into one data stream. So you're right. You can have you can do the Mehe itch by incorrectly analyzing data, and it's usually coming from the point of view of somebody who has a highly supported clade, and they say, oh my goodness, this is great, now I can name something. And I guess the difference is that when I find a new species, what do I say, Michael? When we get a new species, what do? Oh shit! That's the last thing in the world I want to do, is have to go through the pain and bother of describing a new species. But I always do it because that's important and I have to do it. But it's not what I want to do. I have to be dragged like kicking and screaming into naming a new species, because it is too much work, and it's not why I went into ecology, to name new species. So but I think there's a lot of people on the DNA side of things who are just as fallible, and they have their own ways of making mistakes to do it. And yeah.